So, if you spill cancer-causing industrial toxins on the ground, say, a massive petrochemical leak that's just totally saturated with persistent organic pollutants,
Yeah.
traditional environmental science basically gives you, well, one brute force option.
Yeah, you essentially have to dig up the contaminated dirt with heavy machinery.
Right.
You load it into trucks and you incinerate it at incredibly high temperatures. I mean, it's massively expensive.
And super carbon intensive, honestly, a logistical nightmare.
Oh, absolutely. But nature's solution to that exact same chemical problem looks entirely different. It looks like a solitary, unassuming little brown mushroom wearing a tight, white, zigzagging sweater.
Which completely upends our whole human-centric view of problem solving, Like, we look at industrial waste and we just see an unbreakable chemical disaster.
Exactly. But this specific fungus looks at those identical, ultra-complex molecular ring structures, and it basically sees an all-you-can-eat buffet.
And it doesn't just eat them. It obliterates them using an arsenal of what are essentially like remote-controlled molecular drones.
It really does.
So today, we are doing a deep dive into this biological wrecking ball known to science as Hypholoma marginatum, or by its much more charismatic colloquial name, the Snakeskin Brownie.
It's a great name.
Our mission today is to take you inside the microscopic warfare happening right under your boots in a typical pine forest. We are pulling from this incredibly dense stack of materials today.
You've got a lot to cover.
Yeah, we've got a definitive mycological monograph called Evolving Perspectives on Hypholoma marginatum, some cutting-edge biotechnology papers on fungal mycoremediation, and regional biodiversity atlases spanning all the way from British Columbia to the UK.
Plus some biochemical analyses of some incredibly potent secondary metabolites.
Right. And when you pull all these threads together, the portrait that emerges isn't just, a simple forest decomposer.
Not at all. You are looking at a master of physical disguise.
Yeah.
I mean, an organism that uses invisible biofluorescent light to manipulate insect populations.
Which is just wild.
And a species that is currently undergoing an extreme, real-time evolutionary identity crisis right in front of modern genomic scientists.
But to really appreciate how this fungus can theoretically vaporize coal mining waste or, like, deadly herbicides, we have to start with the physical organism itself, because even its name is basically a brilliant biological optical illusion.
It is. If you go back to the earliest taxonomic records, this mushroom's naming history is basically a snapshot of how human beings figured out how to categorize the natural world.
Yeah, the first scientific documentation we have goes back to, what, 1796?
Right, 1796, courtesy of a mycologist named Christian Hendrik Persoon. And, you have to imagine the state of science at the end of the 18th century.
No DNA sequencing, right?
Exactly. I mean, Carl Linnaeus had recently revolutionized biology with his binomial naming system, giving everything a genus and a species. But for fungi, the tools were incredibly primitive.
No electron microscopes either.
No, mycologists like Persoon were literally just wandering through the woods, picking up mushrooms, looking at them with the naked eye, and grouping them by basic macroscopic shapes.
So if a mushroom had, like, a cap, a stem, and gills underneath, it basically just got thrown into one massive junk drawer category.
Yeah, that category was the genus Agaricus. So Persoon found our snakeskin brownie and named it Agaricus marginatus.
And that specific epithet, marginatus, that was based on what he saw on the cap, right?
Yes, his observation of the cap's margin, or its edge, because when the mushroom is young, it has this delicate, silky white partial veil that stretches from the stem to the rim of the cap.
Right, to protect the developing gills.
Exactly, and as the cap opens, that veil tears, leaving a distinct fringed border margin.
But putting thousands of completely different mushrooms into the agaricus bucket obviously wasn't sustainable as the science progressed,
No, it became a mess. So by 1889, a German mycologist named Joseph Schroeder took a much closer look at its anatomy and moved it to a new genus Hypholoma.
And the etymology of Hypholoma captures the physical nature of the organism perfectly, I think. It derives from Greek, and it translates to mushrooms with threads.
Because Schroeder wasn't just looking at the top of the mushroom, he was looking at the entire architecture.
Yeah, and that thread reference works on two very distinct levels. You have those wispy, thread-like remnants of the partial veil hanging off the cap margin.
Right.
But more importantly, if you brush away the pine needles and dig into the dirt at the base of the stem, you find these dense, radiating bundles called rhizomorphs.
They look like thick, twisting, yellowish threads.
Yeah, it's mycelium that has basically bundled itself together into these root-like cords to anchor the mushroom into the decaying wood and transport water over longer distances.
Those rhizomorphs are basically the fungus's supply lines, so Schroeder's reclassification to Hypholoma marginatum gave us a highly accurate anatomical description.
But the colloquial name, the snakeskin brownie, comes from a physical feature on the stipe or the stem that is, well, entirely deceptive.
Completely deceptive.
Let's paint the visual for the listener here. So you're walking through a spruce plantation. You spot a mushroom with a stem about, say, 5 to 10 centimeters long. Near the cap, it's a light ochre, but as you move down toward the base, it turns into this deep, dark, reddish-brown color. And wrapped around that dark, fibrous stem are these brilliant, white, zigzagging bands. I mean, it looks exactly like the geometric scales on a snake's belly.
It really does. It looks like the mushroom is intentionally producing a white pigment to draw racing stripes on itself.
Which is the optical illusion.
Yeah.
Because the fungus is expending absolutely zero energy, producing a white pigment for that pattern.
Yeah, zero. The snakeskin design is an entirely structural byproduct. It's caused by a mechanical failure of the tissue.
It's an issue of differential expansion rates. The way I picture it is like a fast-growing teenager who hits a massive growth spurt in the middle of winter.
Oh, that's a great analogy.
Right. They shoot up 4 inches in a month, but they are stuck wearing this tight, non-stretchy wool sweater.
Right, and eventually the sheer mechanical force of their growing body is going to cause that sweater to burst at the seams.
Exactly. You'd be left with ripped bands of wool hanging off their arms and torso, exposing the skin underneath.
That captures the physics of it perfectly. In this case, the teenager is the tissue of the stipe itself, which is incredibly tough, highly fibrous, and elongates very rapidly.
And the wool sweater is that silky white partial veil we talked about earlier.
Exactly, the tissue that originally protected the gills. As the tough stem rockets upward, the delicate, friable veil tissue clings to it. But the veil lacks elasticity.
It can't stretch.
Right, it cannot stretch to accommodate the growth.
So it basically just fractures. It rips laterally along the stem in these transverse, zigzagging ruptures.
And those white snakeskin bands are just the shredded remnants of that tight sweater.
While the dark negative space between them is the actual underlying tissue of the stem being exposed.
And that underlying stem tissue has to be structurally dense. The literature frequently references the toughness protection hypothesis to explain this.
Right, to explain why the snakeskin brownie invests so much biological energy into building such a rigid, fibrous fortress of a stem.
Because when we normally think of mushrooms, we generally think of them as soft, fleshy, almost fragile things that bruise easily.
Right, like something you'd buy at the grocery store.
Exactly. But this specific fungus lives in a brutal microclimate. The floor of a dense, mature, coniferous forest, whether it's a Sitka spruce grove in the UK or a Russian pine stand, is a surprisingly hostile place for a moisture-dependent organism.
It really is an environment of extremes. You have a forest floor buried in a thick, loose layer of dead needles.
Right, and unlike the dense, damp, composting leaf litter of a deciduous forest, conifer needles are rigid and loosely packed.
They allow the wind to whip right through the understory.
Yeah, and sun patches that break through the canopy can heat up and desiccate the surface layer of needles in a matter of hours. I mean, the evaporation rates are intense.
So if a mushroom wants to survive long enough to mature its spores and release them, it can't afford a soft, watery stem that will just wither and collapse the second a dry wind hits it.
No, it would die immediately. The fibrous density of the stipe is a direct evolutionary adaptation to prevent catastrophic dehydration. It locks the moisture inside.
So the stem is the fortress. But the pileus, the cap of the mushroom, exhibits a completely different survival strategy, doesn't it?
It does. It is characterized by extreme morphological plasticity. You cannot rely on a static description of its shape or color to identify it in the wild.
Because it basically morphs. The cap starts out convex, like a tight little dome, and as it matures, it expands outward until it is completely plain, completely flat.
Usually spanning somewhere between 4 to 9 centimeters across.
Right. And the center might start out as a vibrant brick red, feeding toward the edges, but the color is a total chameleon act because the tissue is highly hygrophanous.
Right, meaning the cells of the cap actively change their optical properties based on their internal hydration levels.
Which is fascinating.
It is. When the tissue is fully saturated with water after a heavy rain, it might appear dark, rich and almost translucent.
But then as that harsh forest wind dries it out, the cells shrink, the light refracts differently, and the cap can fade to a dull yellowish or a muted tan or even a drab gray-brown.
Which is exactly why relying on cap color to ID this mushroom is a fool's errand. You have to look for the ripped sweater stem.
Right, and then you have to look under the cap at the gills because the chromatic shift there is wild.
Oh, the gills are amazing.
They start off as a pale cream color when the mushroom is young. Then they shift to an olive green.
And finally, as the microscopic ellipsoidal spores mature and pack into the tissue, the gills turn a dark, bruised, purplish brown.
And if you take a spore print, like by letting the cap rest on a piece of paper overnight, you get a thick deposit of that deep purple brown powder.
But the visible color changes of the gills are really only half the story. The regional biodiversity research highlights a phenomenon observed in closely related Hypholoma species that completely alters our understanding of how these fungi interact with their environment.
You're talking about the UV light.
Yes. Under ultraviolet light, the hymenium, the spore-bearing tissue of the gills, has been shown to exhibit biofluorescence.
Right, so certain compounds in the gill tissue absorb invisible UV radiation from the environment, excite their electrons, and then re-emit that energy as a visible glowing light.
Yeah, exactly.
But, okay, let me push back on this for a second. From a purely evolutionary standpoint, nature doesn't waste energy for fun, right?
No, never.
Producing complex biofluorescent compounds requires a serious metabolic investment. So if this glow is completely invisible to the human eye, and likely invisible to a lot of foraging mammals, why bother? I mean, it feels like a biological party trick playing to an empty room.
Well, the room isn't empty, we just aren't the intended audience. Human photoreceptors are incredibly limited, but consider the visual systems of the primary inhabitants of that needle-strewn floor. Nocturnal fortis flies, gnats, and specialized beetles.
Oh, right.
The compound eyes of many of these insects are highly tuned to perceive wavelengths deep into the UV spectrum.
So in the deep, shadowy twilight of the conifer understory, where the heavy canopy blocks out most visible light, those gills are essentially acting like a glowing neon diner sign on a dark highway.
Precisely. It is a targeted high-frequency advertisement. The fungus isn't just relying on random wind currents to carry its spores away.
Because wind struggles to penetrate that dense, tangled layer of pine litter.
Exactly. By broadcasting a secret, glowing visual language, the fungus actively recruits insect couriers. The beetles and flies are drawn to the fluorescent beacon, they land on the gills, get coated in thousands of those sticky, purple-brown ellipsoidal spores, and then fly off.
Delivering the spores to new, uncolonized microhabitats far beyond the reach of the wind. That is basically an invisible transit network.
It really is.
So just to recap, the snakeskin brownie has built a fibrous fortress to survive the drying winds. It uses structural illusions and agrafenous color shifting to adapt to the climate, and it's potentially using a glowing secret language to hire insect pilots.
Right.
But surviving the physical elements of the forest floor is just the prelude. The real biological marvel is how this organism secures its food. Because surviving on a diet of dead conifer needles is functionally impossible for the vast majority of life on Earth.
It is. To understand the biochemistry, you really have to frame the ecological niche. The snakeskin brownie is an obligate specialist of the coniferous biome.
Right. The regional atlases from the UK explicitly link it to Sitka spruce plantations. And in the vast taiga of Russia, it is deeply associated with coastal spruce debris.
It doesn't just tolerate conifer litter, it requires it.
And the biodiversity literature notes that finding a healthy population of snakeskin brownies is a prime indicator of forest ectotrophic stability, or FES.
Yes, FES.
Which, when I first saw that term in the sources, I thought, okay, this sounds like really dense forestry jargon. But it's actually describing this massive, invisible tug-of-war happening beneath the soil, isn't it?
Exactly. FES is the ultimate metric of a balanced forest ecosystem. A mature forest operates on two massive parallel fungal networks. First, you have the mycorrhizal fungi.
The symbiotic partners.
Right. These are the fungi that physically fuse with the living roots of the pine and spruce trees. They act as microscopic root extensions, mining the deep soil for water and vital minerals like phosphorus.
And they trade those to the tree in exchange for liquid carbon, right?
Right.
The sugars the tree produces through photosynthesis.
Exactly. They are the engines of growth. But growth cannot happen in a vacuum. If a forest only had mycorrhizal fungi, the trees would eventually pull all the available nutrients out of the soil.
And the dead needles and fallen branches would just pile up hundreds of feet high.
Yes, the entire ecosystem would literally choke to death on its own debris. Which brings us to the second network, the saprophytic fungi.
The recyclers.
The recyclers. The organisms that possess the enzymatic keys to break down dead organic matter and release the locked-up nitrogen, carbon, and trace minerals back into the soil matrix.
So when a forest has high ectotrophic stability, it means these two networks, the growers and the recyclers, are in perfect equilibrium.
And the snakeskin brownie is a premier player in that saprophytic network.
The sources mention that it's absolutely vital for the recovery of log timber areas, because when human logging operations move through a forest, they harvest the massive trunks but leave behind tons of what they call slash.
Right, the branches, the bark, the millions of acidic needles. The ecosystem is suddenly buried in a chaotic, unmanageable volume of dead matter.
So the snakeskin brownie acts as a first responder. It rapidly colonizes the slash to kickstart the nutrient cycle so the forest can begin to heal and regenerate.
But processing that slash requires an incredibly specialized digestive system. A conifer needle is not a soft, welcoming environment.
No, it's highly acidic. It frequently drops the local pH to levels that would denature most proteins.
And it is packed with toxic, antimicrobial resins designed to repel insects and bacteria while the needle was alive.
And most formidably, the vast majority of the needle's caloric value is locked behind an impenetrable fortress of lignin.
Lignin is incredibly tough.
Yeah, if cellulose is the sweet, energetic filling of a plant cell, lignin is the bulletproof glass protecting it. It is essentially the plant kingdom's version of synthetic plastic.
Chemically, lignin is this massive, chaotic, three-dimensional web of tightly bonded aromatic carbon rings. It is incredibly heavy, incredibly dense, and completely insoluble in water.
Right, so if a standard soil bacterium bumps into a lignin molecule, it literally doesn't have the biological tools to even scratch the surface. The bacteria just bounce off.
This is why the snakeskin brownie is classified functionally as a white rot fungus, or more specifically in this habitat, a litter decomposing fungus, or LDF.
Right. And white rot fungi are basically the elite demolition crews of the biosphere. They are the only organisms on Earth that have evolved the comprehensive enzymatic toolkit required to completely shatter the three-dimensional architecture of lignin.
And the undisputed heavy artillery in the snakeskin brownie's chemical arsenal is an enzyme called manganese peroxidase, usually abbreviated as MNP.
The biochemical analyses in our sources map out the specific stats of this enzyme, and honestly, it operates like a microscopic, highly calibrated piece of military hardware.
It really does. MNP is a heme-containing protein, meaning it has an iron core similar to the hemoglobin in our own blood.
Which allows it to facilitate complex oxidation reactions.
Exactly. But the physical dimensions and stability metrics of the MNP produced by the Strophareaceae family, the family containing the snakeskin brownie, are what make it so devastatingly effective. First, it has a very specific molecular weight.
Roughly 41 to 42 kilodaltons, right?
Yes. And to put that in perspective, a kilodalton is a unit of atomic mass used to measure proteins. A protein of 41 kilodaltons is relatively compact.
So if the fungus produced a massive, bulky enzyme, say 100 kilodaltons, it would be utterly useless. Because the microscopic pores and crevices inside a decaying pine needle or a piece of woody debris are incredibly tight.
Exactly. A bulky enzyme would just get stuck on the surface. The 41 kilodalton size means the MNP can physically infiltrate deep into the cellular structure of the wood before it detonates.
It's perfectly sized for infiltration, and once it's inside, it has to survive the local chemistry. Which brings us to its near-neutral isoelectric point, or PI.
Which the research clocks between 6.3 and 7.1.
Right, and an isoelectric point is essentially the specific pH level where a molecule carries zero net electrical charge. I think of it like a submarine trying to maintain neutral buoyancy in an ocean of varying densities.
That's a good way to picture it.
Because the forest floor is completely chaotic chemically.
Yeah.
1 inch of soil might be highly acidic because a pocket of pine resin is decaying, while an inch away it might be totally neutral.
Right. And if an enzyme's isoelectric point is too extreme, the fluctuating pH of the forest floor would rip its electrical bonds apart. The protein would denature and unfold, rendering it useless.
But by holding a near-neutral isoelectric point, the MNP remains structurally stable and fully armed across a wildly unpredictable environment.
So we have a stable, deeply infiltrating enzyme. But the true genius of manganese peroxidase is how it actually attacks the lignin. Most digestive enzymes work through direct contact.
Like a human stomach enzyme binds directly to a protein molecule to break it apart.
Exactly, but lignin is too massive, and its carbon bonds are too recalcitrant for a simple lock-and-key binding mechanism.
So the fungus can't chew the lignin, so instead it uses the MNP to launch remote-controlled drones, which is just a brilliantly elegant chain reaction.
It is. The enzyme sits inside the fungal tissue and pulls in raw, ambient manganese 2+ ions from the surrounding soil. Then, using hydrogen peroxide, which the fungus naturally generates as an internal metabolic byproduct, the MNP catalyzes an oxidation reaction. It strips an electron away from the manganese 2+, converting it into manganese 3+.
And manganese 3+ is a highly unstable, intensely reactive ion, right? It desperately wants that missing electron back.
Exactly. But the enzyme doesn't hold on to it. The Mn3+ ions act as diffusible redox mediators.
So they are the drones. The highly reactive Mn3+ ions diffuse away from the fungal body, traveling microscopic distances deep into the dense wood matrix.
Yes.
They physically bypass the tight pores, eventually colliding with the massive plastic-like lignin structures. And because the Mn3+ is so desperate for an electron, it literally rips one out of the lignin molecule's aromatic rings.
And when you violently strip electrons away from a stable polymer like that, the chemical bonds just can't hold together.
The lignin structure destabilizes, cracks, and shatters into smaller, disjointed pieces. It's like the fungus is digesting a microscopic fortress from the inside out without ever actually touching it.
It is literal remote-controlled chemical warfare. And the MNP drones are supported by an even more aggressive, non-specific secondary attack system known as the Fenton reaction.
Oh, the Fenton reaction.
Yeah.
This is pure biological violence. Instead of manganese, this reaction utilizes iron, right?
Yes. The fungus pumps out iron 2+ ions and reacts them with more of its own hydrogen peroxide in the extracellular space.
And when those two molecules collide, the resulting chemical reaction violently ejects what are called hydroxyl radicals.
Right. A hydroxyl radical is essentially an oxygen atom paired with a hydrogen atom, but it carries an unpaired electron. In biological systems, hydroxyl radicals are among the most aggressive, indiscriminately destructive oxidants in existence.
So if the Mn3+ ions are the targeted, remote-controlled drones seeking out lignin, the hydroxyl radicals produced by the Fenton reaction are essentially millions of microscopic, wildly spinning chemical buzzsaws.
That is exactly what they are. They fly through the wood matrix and just snip the chemical chains of absolutely anything they bump into.
They don't care if it's lignin, if it's the structural hemicellulose, or the energy-rich cellulose itself. They just cleave carbon bonds indiscriminately, softening the entire woody structure into a chemically fragmented slurry.
But the snakeskin brownie isn't just mindlessly blowing up the wood. White rot fungi engage in a highly choreographed metabolic sequence called preferential delignification.
Right, they exert massive control over this chaotic chemistry. In the early stages of decay, they prioritize the deployment of the MMP drones to specifically target and shatter the lignin shield, while actively suppressing the degradation of the softer cellulose.
They obliterate the armor but leave the prize intact.
The result is incredible. If you've ever kicked open a rotting log in a pine forest and seen that bright, white, stringy, almost sponge-like wood inside, that is the aftermath of preferential delignification.
The fungus has chemically vaporized the dark, dense lignin, leaving behind a bleached, fibrous residue that is incredibly rich in easily digestible cellulose.
They essentially unlock the biological pantry, consume what they need, and simultaneously create a nutrient-dense feeding ground for hundreds of other soil bacteria and insects that follow in their wake.
And to manage all of this without exhausting itself, the fungus's biochemical factory is incredibly frugal. It does not pump out these expensive enzymes 204/7.
Right. The research highlights that the production of manganese peroxidase is highly regulated. It is strongly enhanced only by the physical presence of environmental manganese.
The fungus literally senses its environment. It tastes the soil, detects the raw metal it needs to build its drones, and only then does it flip the switch to kick the enzyme factory into high gear.
It's a masterclass in metabolic efficiency. But this is the exact point where the story of the snakeskin brownie leaps out of the realm of obscure forest ecology and collides directly with human history.
Yes, the biotechnology applications.
Because if this fungus has evolved this incredibly complex or highly efficient system to shatter the unbreakable three-dimensional aromatic rings of natural lignin using remote chemical drones, well, biotechnology researchers started asking a revolutionary question. What happens if this little brown mushroom bumps into complex, man-made chemicals that share a similar structural blueprint to lignin?
And the answer to that question forms the foundation of mycoremediation science. Because the fungus does not differentiate between a complex natural polymer and a complex synthetic toxin.
It treats our most lethal, persistent industrial pollutants exactly like it treats a tough pine needle. It eats them.
It does. Let's look at the real-world application of this. The mycoremediation literature heavily focuses on the fungus's ability to mineralize PAHs.
For anyone who hasn't worked in environmental toxicology, PAH stands for polycyclic aromatic hydrocarbons. These are some of the most notorious, highly regulated environmental pollutants on the planet.
A classic example of a PAH is benzoapyrenene. These compounds are the heavy, toxic byproducts of industrialization.
You find them saturating the soil around coal mining operations, crude oil spills, heavy petrochemical processing plants, and even in the ash of incinerated waste.
They are highly potent carcinogens. And what makes them so devastating to an ecosystem is their molecular architecture.
Their structure is the problem. Polycyclic aromatic means their chemical backbone is constructed from multiple fused rings of carbon. It is an incredibly dense, ultra-stable molecular configuration.
It is the exact same underlying geometry as natural lignin. This is why PAH are classified as persistent.
Right. Standard soil bacteria encounter a clump of benzo to pyrene, try to digest it, and fail completely because they don't have the tools to crack the aromatic rings. So the toxin just sits in the dirt for decades, leaching into the groundwater and poisoning the food web.
But when a species from the Strophaceae family, like the snakeskin brownie, encounters that same contaminated soil, it initiates its standard foraging behavior.
It secretes its manganese peroxidase, oxidizes the ambient manganese, and sends those highly reactive Mn3+ chemical drones out into the soil matrix.
The drones collide with the cancer-causing PAH molecules. And because the drones are just looking to violently strip electrons away from aromatic carbon rings, they rip the benzopyrene apart just as easily as they would a piece of dead wood.
And the laboratory screenings detailed in the sources show that these fungi don't just absorb or quarantine the toxins, they achieve complete mineralization.
Mineralization is the ultimate goal of remediation. It means the complex, toxic molecule is completely dismantled at the atomic level, broken down through successive oxidation reactions until all that remains is harmless carbon dioxide gas and basic, non-toxic, water-soluble fragments.
That can be safely absorbed by the soil ecosystem, so the toxicity is literally erased from existence.
Yes, and because researchers understand the biological triggers of the fungus, they found a massive chemical hack to accelerate the cleanup.
Right, because we know the fungus only ramps up MNP production when it detects manganese in the environment. So in the laboratory trials, scientists deliberately supplemented the PAH-polluted soil with extra manganese.
And the results were staggering. By simply providing the fungus with an abundance of the raw material it uses to build its weapons, the degradation rate of those deadly carcinogens jumped 20-fold.
Wow, 20-fold?
Yeah. You feed the mushroom a little extra metal, and its enzyme factory goes into extreme overdrive, shredding the industrial waste 20 times faster than it normally would.
That is just a breathtaking biological exploit. And the enzymatic flexibility doesn't end with coal tar and oil spills either. The biochemical arsenal is perfectly suited to attack modern agricultural toxins as well.
Right, the herbicide studies.
Yeah, specific degradation studies zeroed in on Hypholoma dispersum, which the taxonomic data confirms is a widely accepted synonym for our snakeskin brownie, Hypholoma marginatum. Researchers placed the fungus in environments contaminated with lethal persistent herbicides, specifically paraquat and glyphosate.
Now, paraquat is an incredibly aggressive contact herbicide. It is highly toxic to humans and mammals, causing severe, irreversible lung damage if inhaled or ingested.
And it is notorious for binding tightly to soil particles and resisting natural degradation, basically rendering agricultural land toxic for extended periods.
Yet, when the snakeskin brownie was introduced to the contaminated substrate, the studies showed it efficiently degraded up to 54.1% of the total paraquat volume in a matter of days.
To contextualize how impressive that is, the researchers ran parallel tests using specialized bacterial remediators, specifically Pseudomonas perdita.
Yes, a bacterium that is actively engineered and widely deployed in commercial waste management to clean up chemical spills.
And the little brown mushroom from the pine forest significantly outperformed the industrial bacteria.
It really highlights a paradigm shift in how we approach environmental disaster zones. The traditional methods, excavation, chemical washing, high-temperature incineration, are not only prohibitively expensive, but they effectively sterilize the soil.
Right, they kill off the entire microbiome in the process of cleaning it. But bioremediation offers an in situ, biologically regenerative alternative.
But taking it from a pristine laboratory petri dish into the real world is incredibly complex.
Yeah, I have to ask, are we saying we could literally sprinkle conifer needles and snakeskin brownie spores over a toxic chemical spill and just let the mushrooms eat it? Like a magic sprinkle?
Well, no, it's not quite a magic sprinkle. You can't just take a dump truck full of needles, mix in a few billion spores and a bag of manganese dust, airdrop it over an oil spill, and expect the pollution to vanish overnight.
Right, because you are dealing with highly sensitive living organisms that have to compete with native bacteria, weather fluctuations, and unpredictable moisture levels.
Exactly. Which is why the field observations detailed in the sources are so critical. Look at the data from Litvinov City in the Czech Republic.
Okay, Live City.
Litvinov is not a controlled laboratory. It is a region carrying the heavy, toxic legacy of intense, long-term industrialization. We are talking about decades of intensive brown coal mining, sprawling chemical processing plants, and heavy petrochemical refineries.
So the urban and peri-urban soils in and around Litvinov City are basically a worst-case scenario for environmental health. The soil profiles are completely saturated with a cocktail of persistent organic pollutants, or POPs.
It's an environmental graveyard of deeply embedded PAHs, polychlorinated biphenyls, PCBs, and highly toxic dioxins. By all conventional biological metrics, the soil should be dead.
But when field mycologists actually surveyed these toxic hotspots, they didn't find sterile, lifeless dirt. They found highly diverse communities of native, saprotrophic fungi actively thriving right in the epicenter of the pollution.
Fungi from families like Agaricaceae, Saccharolaceae, and our lignin-degrading champions, the Strophareaceae.
So the fungi had already moved in. They didn't need human intervention to start the cleanup. Over decades of exposure, these local fungal populations recognized the massive carbon reserves locked inside the industrial pollutants, adapted their enzymatic output, and began living off the elevated concentrations of the toxins.
Nature is already performing microremediation on a massive scale. The goal of modern environmental biotechnology is not to invent a new solution, but to identify these native champion species, sequence their genomes, and understand their precise enzymatic triggers.
But the manganese boost.
Exactly. And optimize the environmental conditions to help them do what they have already evolved to do, just much faster.
It is an incredibly hopeful vision for the future of our planet. The idea that a little brown mushroom quietly eating pine needles possesses the exact chemical machinery required to undo humanity's worst industrial mistakes is staggering.
It really is.
But to be this versatile chemically, to be able to fluidly switch your diet from dead spruce woods to toxic coal tar to deadly agricultural herbicides, an organism has to be characterized by extreme adaptability.
Absolutely.
And as we look at the most recent cutting-edge genetic research on the snakeskin brownie, we realize that its adaptability isn't just chemical. It is actively altering its physical form, its growth habits, and its very DNA in real time.
Yes. This fungus is currently undergoing such a profound identity crisis that it is fracturing the very way scientists classify life.
We are entering the territory of cryptic species complexes and extreme morphological plasticity.
To understand the magnitude of what the geneticists are finding, we first have to establish the baseline behavioral profile of this fungus in the wild.
Right. Let's look at how it normally behaves compared to its relatives. The most famous, widely recognized cousin of the snakeskin brownie is the sulfur tuft, Hypholoma fasciculare.
Yes.
If you have ever walked through the woods and found a dead stump completely smothered in hundreds of lurid, neon yellow mushrooms packed so tightly together they look like a single organism, that's the sulfur tuft.
In mycological terms, that growth habit is described as strongly gaspitose. They fruit in massive, densely packed tufts or clusters, aggressively colonizing a single concentrated food source.
But the snakeskin brownie exhibits the exact opposite behavior. Field guides frequently refer to the lone brownie phenomenon.
It is notoriously solitary. You rarely find two of them touching. You usually find a single specimen standing alone in the pine needles, or perhaps a loosely scattered handful spread out over a wide area in what mycologists call a trooping formation.
It's a very deliberate evolutionary dispersal strategy, isn't it?
It's risk management. The sulfur tuft bets all its reproductive energy on one massive, localized explosion of spores from a single log. The snakeskin brownie spreads its bets.
Because conifer forest floor is chaotic, and resources like buried twigs or optimal moisture pockets are distributed randomly. So popping up singly across a much wider geographic footprint maximizes the mathematical probability that at least a few of its spores will catch a localized wind current and land on a fresh, uncolonized piece of real estate.
Precisely. And that solitary behavior was the established, unquestioned scientific consensus for over a century. If it's clustered, it's a tuft. If it's alone, it's a brownie.
But in 2016, a discovery in the UK completely shattered that assumption.
The Kew Gardens dilemma.
Yeah, the event took place in Wales. Field researchers stumbled upon a population of fungi growing on a massive pile of conifer wood chips. The environment was highly artificial, essentially a hyper-concentrated, easily accessible mountain of high-nutrient food.
Vastly different from the sparse, tough needle litter of a natural forest floor.
And the fungus completely changed its strategy to exploit it. It wasn't solitary. It was growing in massive, dense, suffocating clusters.
It was exhibiting a violently cuspitose growth habit, looking behaviorally identical to a sulfur tuft.
The physical form of the mushroom had warped so radically in response to the sudden nutrient abundance that the expert field mycologists on site couldn't even identify it. It defied every visual diagnostic metric in the textbooks.
It went way past the limits of visual field mycology. They had to send tissue samples to Martin Ainsworth, a senior mycologist at the Royal Botanic Gardens in Kew.
And Ainsworth had to bypass the physical morphology entirely and run a full DNA profile just to figure out what organism they had found.
And the DNA confirmed the impossible. This clustering, totally unrecognizable fungus was 100% Hypholoma marginatum, the snakeskin brownie.
Its morphological plasticity is so extreme that it can basically rewrite its physical growth blueprints on the fly if the environment demands it.
But while the Welsh population changed its shape, its core DNA remained the same. If you cross the Atlantic and look at populations in North America, the identity crisis goes much deeper.
The actual genetic code of the fungus is beginning to splinter.
This brings us to the Pacific Northwest DNA split. For decades, mycologists exploring the deep, temperate rainforests of the Pacific Northwest in the United States and Canada have cataloged what they visually identified as the snakeskin brownie, often logging it under its synonym, Hypholoma dispersum.
But there was a glaring ecological discrepancy, right?
Yes, they weren't eating conifer needles. In Europe and Russia, the fungus is an obligate specialist of the spruce and pine biomes. But in the Pacific Northwest, these identical-looking mushrooms were found thriving on wet sedge, a grass-like plant, and decaying broadleaf debris in riparian zones.
They'd completely abandoned their ancestral diet and habitat.
And when modern genomic tools became widely available, researchers decided to look under the hood. They extracted the DNA from these sedge-eating Pacific populations and sequenced their internal transcribed spacer, or ITS, regions.
Now, the ITS region is effectively the universal genetic barcode for fungi, right?
Yeah.
It's the highly conserved sequence of DNA used to definitively identify and separate species.
Exactly. And the sequencing revealed that the Pacific Northwest populations had a 3% divergence in their ITS DNA compared to the standard pine-eating European H. marginatum.
Okay, to someone outside the field of genetics, 3% sounds like a rounding error. It sounds negligible.
Sure.
But in the rigid math of fungal taxonomy, what does a 3% shift actually represent?
It represents an evolutionary earthquake. In fungal genomics, a 3% divergence in the ITS region is not a minor mutation. It is highly significant. It is frequently cited as the standard threshold required to justify declaring an organism a completely new, independent species. Because of this massive genetic drift, researchers have isolated this specific North American population and given it a provisional designation. Hypholoma dispersum PNW04.
We are literally watching evolutionary radiation happen in real time. The fungus migrated across continents, found a completely alien food source, wet sedge, instead of tough pine needles, adapted its digestive enzymes to survive, and over time, its very genetic blueprint has drifted away from the European original.
Yes.
But this triggers a massive philosophical dilemma for science. If it acts differently, eats differently, and has different DNA, at what point do we stop calling it a snakeskin brownie and admit it's a completely different organism? At what point do we stop trying to force it into the historical 18th century box created by Christian Pursoon?
You've just articulated the central tension in modern biology. It is a clash between traditional field mycology, which relies on tangible, observable traits, what we can see and touch in the forest, and modern genomics, which reveals the invisible underlying code. We are staring at what evolutionary biologists call a cryptic species complex.
a cryptic species complex, meaning a group of distinct species that have evolved to look absolutely identical to human eyes, hiding their genetic differences behind a shared physical disguise.
Exactly. As humans, we possess a deep psychological compulsion to categorize the natural world. We want neat, tidy filing cabinets. We want to point at a mushroom, give it one definitive Latin name, and be done with it.
But nature is not a filing cabinet. It is a fluid, incredibly messy, continuous spectrum of adaptation.
Right. The genomic reality of the snakeskin brownie forces us to confront the limitations of our own classification systems. Is it one global superspecies with unimaginable adaptability, or is it a sprawling family of hyperlocal sibling species, each perfectly tailored to its specific microenvironment?
It really redefines what the word species even means. But while debating the philosophical boundaries of DNA is fascinating in a sterile laboratory, confusing the snakeskin brownie with the other species sharing its habitat is not an academic exercise if you're actually out walking in the woods.
No, it's absolutely not.
Because as we examine the final pieces of source material, we have to look at the dark side of the forest floor. The chemical arsenal of this fungus is not limited to digesting wood and industrial waste.
The family is steeped in chemical warfare.
And making a visual misidentification in the field can easily be a fatal error for a human being.
The stakes for foraging in these specific habitats are literally life and death. The snakeskin brownie itself, H. marginatum, is universally classified as inedible. While it won't necessarily kill you, it is considered mildly nauseogenic, and its tissue is packed with a complex array of highly bioactive secondary metabolites that make it profoundly unpalatable.
Let's look at those secondary metabolites, or SMs. The name snakeskin brownie makes it sound like a benign, rustic baked good. But a chemical breakdown of its tissue reads like a pharmaceutical catalog.
It does. The Hypholoma genus is a prolific synthesizer of complex chemistry. They are rich in terpenoids and alkaloids. Specifically, many of these species actively manufacture nitrogen-containing indole derivatives.
And these specific alkaloids are the compounds responsible for giving the fruiting bodies an intensely bitter, acrid, completely repulsive taste.
Yes, and it produces dictopyperazines, which are cyclic dipeptides. Interestingly, medical researchers are actually mining these specific compounds right now because they show significant promise in laboratory trials as both antimicrobial agents and potential anti-cancer drugs.
Combine that with the high concentration of beta-glucans, complex polysaccharides known to aggressively modulate and stimulate the immune system, and you have a biologically potent cocktail.
Very potent. But the warfare isn't just inside the mushroom. The sources detail how the fungus actively alters the soil chemistry around its mycelial network to sabotage its neighbors. It secretes oxalic acid directly into the dirt.
Which is a brilliant territorial defense mechanism. By pumping oxalic acid into the surrounding matrix, the fungus deliberately crashes the local pH, making the soil hyperacidic.
And most competing soil bacteria require a relatively neutral pH to reproduce and thrive. The acid bath effectively sterilizes the fungus's immediate territory, suppressing bacterial invasions and ensuring the fungus has exclusive access to the nutrients it is mining.
The forest floor is not a peaceful graveyard. It is an invisible, ruthless fight for calories and space. The secondary metabolites aren't random byproducts. They are an ancient, highly refined arsenal.
The bitter alkaloids deter grazing insects and mammals. The dictopyperazines act as localized antibiotics, and the acid controls the soil.
But some of the organisms sharing this exact same habitat have escalated that chemical deterrence to a lethal extreme. And this is where the morphological plasticity of the snakeskin brownie becomes genuinely dangerous.
Oh, the lookalikes.
Because growing right next to it in the conifer needles is a deadly lookalike known as Gallerina marginata, appropriately and terrifyingly named the funeral bell.
Gallerina marginata is a species that commands absolute, uncompromising respect from every field mycologist. The risk of tragic confusion is incredibly high because their ecological overlaps are almost identical. Both the snakeskin brownie and the funeral bell favor decaying coniferous wood. Both emerge from the exact same needle litter.
And both feature a generally convex to plane cap that falls in the brown to yellowish tan color spectrum, especially when dried out by the wind.
Right. If you are casually scanning the forest floor, spotting a brown cap popping through the pine needles, your brain can easily conflate the two.
But if you make that mistake and you decide to harvest and consume it, the consequences are catastrophic. Because the funeral bell doesn't just use bitter alkaloids to deter predators. Its tissues are saturated with amatoxins.
Amatoxins are some of the most formidable natural poisons on Earth. They are the exact same class of lethal cyclopeptides found in the infamous death cap mushroom, Amanita phalloides.
Let's break down exactly what an amatoxin does to the human body, because it's not just a severe stomachache, it is a fundamental cellular shutdown.
The mechanism of toxicity is chillingly precise. When ingested, amatoxins travel through the bloodstream and target the liver and kidneys. Once inside the cells, they act as highly potent inhibitors of an enzyme called RNA polymerase II.
RNA polymerase II is basically the engine of cellular life, isn't it?
It is. It is the enzyme responsible for reading your DNA blueprints and transcribing them into messenger RNA, which the cell then uses to build the proteins it needs to survive, repair itself, and function.
So when the amatoxin binds to that enzyme, the transcription process instantly stops. The cellular factory is forcefully shut down. The cell can no longer synthesize proteins, and without new proteins, the cell simply starves and undergoes necrosis. It dies.
Because the liver absorbs the brunt of the toxins from the digestive tract, this leads to massive cascading hepatic necrosis. In simple terms, it causes catastrophic, irreversible liver failure, followed rapidly by kidney failure.
And consumption of a surprisingly small amount of Hypholoma marginatum is frequently fatal without a highly invasive rapid organ transplant.
Which makes accurate identification in the field the literal difference between life and death.
So if cap color and habitat are identical, how do you reliably separate the harmless brownie from the lethal bell?
It requires meticulous, granular observation of the physical structures we discussed earlier, the stipe and the spores. The funeral bell typically features a much more uniform, smooth, brownish stem. Crucially, it often retains a distinct, true ring on the upper stem.
A delicate skirt of tissue left over from its own veil, though it can be fragile and ephemeral.
Whereas the snakeskin brownie lacks that distinct ring and instead has that unmistakable, fibrous, deeply tough stem covered in the shredded, white, zigzagging veil remnants.
The ripped sweater pattern.
Exactly. But what if a heavy rainstorm washes away the veil remnants or an insect chews off the ring of the funeral bell?
And what?
If the structural clues on the stem are compromised, you must look at the spores. You perform a spore print. The deadly funeral bell will deposit a thick, distinctively rusty brown spore print.
Oh, whereas the snakeskin brownie, as we established, deposits a dark, bruised, purple-brown print. That subtle shift in the color palette of microscopic spores is the definitive diagnostic marker.
Yes. And the chemical minefield doesn't stop with the funeral bell. Even within its own genus, the snakeskin brownie is surrounded by violently toxic cousins. We talked earlier about the sulfur tuft Hypholoma fasciculare with its massive, clustering growth habit.
Right. And while it lacks amatoxins, the sulfur tuft is violently poisonous in its own right. Its tissue contains a suite of compounds known as fascicularones.
Ingestion triggers severe, debilitating gastric distress, intense vomiting, and there are documented medical case studies where consumption resulted in temporary paralysis and fulminant hepatitis.
Which is an acute, incredibly severe inflammation and rapid failure of the liver. And to make matters worse, there is yet another cousin, the conifer tuft, Hypholoma capnoides. It grows on the same wood in the same clusters.
And some very brave, highly experienced foragers actually consider the conifer tuft to be a choice edible mushroom. But it looks so remarkably similar to the liver-destroying sulfur tuft that actively foraging for it feels like playing Russian roulette with your internal organs.
It's amazing that a mushroom so skilled at cleaning up human toxins is surrounded by cousins that are basically bioweapons aimed at us. I mean, it's a profound, dark irony. We spent the first half of this deep dive marveling at a mushroom that possesses the exact enzymatic tools required to literally clean up humanity's worst, most carcinogenic industrial toxins.
It is a biological purifier.
Yet it lives in a habitat surrounded by cousins and lookalikes that are essentially microscopic bioweapons aimed directly at our livers and kidneys.
It is a stark irony, but it perfectly illustrates the central truth of evolutionary biology. We have to decenter ourselves from the narrative. These incredible secondary metabolites, the amatoxins, the fascicularones, the complex alkaloids, were not designed to punish human foraging.
They weren't meant for us.
No, fungi have been refining these chemical weapons for hundreds of millions of years, locked in a brutal arms race on the forest floor long before primates even evolved. They are designed to dissolve competing nematodes, to hold territory against aggressive soil bacteria, and to paralyze ancient lineages of fungivorous insects.
When a human being misidentifies a mushroom, eats it, and suffers fulminant hepatitis, our reaction isn't the primary goal of the toxin. We are simply biological collateral damage.
Exactly. We are blindly stumbling into an ancient, microscopic war zone that has absolutely nothing to do with us.
That is an incredibly humbling perspective. We aren't the target. We're just collateral damage in a billion-year war over the calories locked inside dead pine needles. So what does this all mean? If we step back and look at the entirety of the source material we've unpacked today, the sheer scale of what is happening inside this single organism is breathtaking.
It really is. We started by looking at what most people would dismiss as an obscure, completely unremarkable little brown mushroom growing in the dirt.
But by peeling back the layers, the taxonomic history, the structural morphology, the complex lignin-degrading biochemistry, and the cutting-edge genomic sequencing, a completely different organism emerges.
The snakeskin brownie is a marvel of evolutionary engineering. It is a highly specialized survivor that utilizes a brilliant structural illusion on its fibrous stem to survive the dehydrating winds of the conifer understory.
It doesn't just digest its food. It deploys an arsenal of remote-controlled chemical drones, manganese 3+ ions, and wildly destructive hydroxyl radical buzzsaws to remotely shatter the unbreakable plastic-like lignin polymers of the forest.
And by utilizing that exact same ancient chemistry, it holds the astonishing, globally significant potential to eat our worst industrial messes, mineralizing cancer-causing polycyclic aromatic hydrocarbons and deadly agricultural herbicides into harmless water and carbon dioxide.
And perhaps most remarkably, it is so wildly adaptable that it is refusing to stay in the taxonomic boxes we build for it. It is actively evolving, splintering its DNA, and radiating into new cryptic species right under our noses in the wet sedge of the Pacific Northwest.
It is a reminder that the quietest corners of the natural world are often the most radically advanced.
It really is. But I want to leave you, the listener, with a final lingering thought. Something to mull over the next time you are walking through the dark, quiet, needle-strewn understory of a mature pine forest. We know that the genetic code of the snakeskin brownie is splintering. We know that the Pacific Northwest clade, PNW04, is actively moving into entirely new territory, adapting to a completely alien diet of wet sedge.
It is expanding its empire.
But to do that, it has to physically transport its spores out of the dense, wind-blocking conifer litter and into those new environments. Earlier, we discussed the biofluorescence observed in the gills of closely related Hypholoma species. Researchers are actively pushing to test the snakeskin brownie for those exact same UV glowing compounds.
Yes, the glowing gills.
So think about this. What if those potentially glowing gills aren't just a quirk of chemistry? What if they hold the secret to its rapid evolutionary radiation? What if, in the dim, shadowy twilight of the forest floor, this fungus isn't just passively waiting for the wind? What if it is actively broadcasting an invisible glowing language to recruit entirely new, highly mobile insect partners?
Essentially hitching a ride on the backs of specialized beetles and flies to travel miles away to completely new alien habitats.
Exactly, allowing it to continue its rapid, aggressive evolutionary journey in the dark, entirely hidden from human eyes.